Hello, this is a talk based on the paper Exploring Brain Transcriptomic Patterns, a topological analysis using spatial expression networks. And this has been accepted as a paper in the Imaging Genomics workshop. Unfortunately, none of us, the authors, was able to attend the conference, and we apologize for that. But we still decided to send the poster and a recording with a very brief explanation on some main topics that are related to the paper. So I'll start with the intro. Our main motivation for this work was, in fact, the recently published paper by Ricciardi, which is correlating gene expressions with functional brain networks. And we had a more detailed look about what has been done in the neuroscience community with respect to the genomic data sets and how they vary in anatomical locations. So essentially what we managed to discover as related work is that first, anatomical locations in the brain vary with respect to their genetic consistency, and in the same time region to region genetic similarity show that there is some association to functional brain function, functional activity, and possibly some other underlying hidden structures which we want to explore. For this reason, we wanted to model the gene's transcription activity with respect to the different anatomical locations. We know that genetic expressions with respect to each gene are over a set of are usually recorded over a set of anatomical locations. However, this vectorial representation misses on the interconnected structure coming from the brain. And as recent studies show that, well, regions which are close to each other are genetically more similar, or with respect to different brain function, different regions have the same genetic structure, we decided that it's very important to actually be able to model the gene with respect to to model the gene's expression values with respect to the different anatomical locations. For this reason, we decided to create a gene spatial expression network. For each gene, the nodes are some defined brain regions of interest, and the edges between nodes are their weight represents the genetic expression similarity between these two brain regions. We applied this procedure to 17 well-expressed genes for which we have some known gene ontology information, and we obtained an ensemble of 17,000 gene networks. And the main problem that we wanted to solve, or the hypothesis that we decided to address, was, well, if we cluster those spatial expression networks, or SENs as we call them, is their topological similarity going to reflect the biological similarity of the corresponding genes? And the main hypothesis is that if clusters of topological similar SENs exist, then the corresponding genes within each cluster may also share similar biological properties. Now, we use the Allen Human Brain Atlas dataset to approach this problem and to address this hypothesis, since up to our knowledge this is the only atlas which has quite detailed transcriptomic information from thousands of brain regions. We had quite stringent selection procedure for the brain regions that we selected. That's why we ended up with 105 brain regions in the left hemisphere. You can see them here. Then, as I said, each SEN, so each spatial expression network for each gene is represented by nodes and edges. The nodes are those 105 brain regions. we define the edges in the following way. Where the edge weight between regions I and J is just 1/1 plus the absolute difference between the mean expression levels of the gene in brain region I and brain region J. The main idea was that we wanted a monotonically decreasing distance, and the larger the differences, the closer the closer the edge weight to 0 is. In this way, we constructed 16,906 spatial expression networks. Now, in order to cluster them, we used a standard graph embedding technique, where we decided to take vectorial representations for in network, and these vectorial representations captured the node degrees. The node degrees are very intuitive, since they represent the local density of each region, and since we had fully connected networks, we knew that the node degrees were bounded between 0 and 105. And for us, it was quite intuitive, because we were interested for each gene and for each region its interconnected activity to all other brain regions. However, this is not the only way that you can define these feature vectors. But for this paper, this is the main feature vector that we used. Then, in order to cluster two networks, we defined the distance between the two networks with respect to the feature vectors by taking the Euclidean distance. We also performed the same procedure with another two feature vectors, a centrality measure for each node. So we defined this feature vector in terms of the centrality score of each node, or we also took the vectorized version of all edge weights, and we explored what the results of using different feature vectors. However, we focused on node degrees only. In order to select a good clustering procedure and number of clusters, we used three different clustering procedures: PAM, C-means, K-means, and we used two permutation procedures. The details of this selection are provided in the paper. We were considering both the stability of the method, we were considering different internal validation measures, we were looking also at the within a cluster we wanted to. At the end, we decided to select three clusters, just to explore whether the broad topological similarity in fact respects some broad biological similarity. We also performed some two-dimensional visualization. We see that there you can see like around three clusters, like two very distinct groups and one group in the middle, which clearly follows some shared topological measures. We have further plots included in the paper. Here we have not included them, but on closer exploration, this cluster has very different topological properties than the genes and the sense in this cluster, while the genes and their sense in the middle cluster have very intermediate values. So as an initial exploration, it makes sense to look at the three big clusters. Okay, detailed explanations of and interpretation of this plot is provided in the paper. However, it's very interesting to actually have a look at this plot, which is very similar to many other plots obtained in different papers in the literature, which shows that there is strong heterogeneity within the frontal cortex. There is some sort of homogeneity in the basal ganglia, but again in the amygdala, in the hippocampus, in the and in different subregions of the basal ganglia, you can see some within heterogeneity and between heterogeneity. The cerebellum, of course, has very strong heterogeneity, and it was just nice to have a look at such a plot composed of all genes that we look to validate that yes, using the sense actually, the SEN networks actually make sense, and yes, they capture similar information. They capture information but in a very different way. But some of the final results are very similar to what has already been found in the literature, and that was very important for us. Then we decided to produce gene ontology analysis. We used the Revigo software, which reduced this long list of significantly enriched geoterms. We got some overrepresented representative terms for each cluster, and we saw that one of the clusters, essentially the turquoise cluster here, was very related to transcription and translation processes. The other cluster, which was very far away, was actually very from the transcription and translation, was actually strongly related to immunity. And then the cluster in the middle, which had intermediate topological structures, it was closely related to the nervous system. So it was very interesting to see that we actually have the topological similarity, which reflects on the biological similarity. We also ran disease enrichment procedure, which confirmed that the genes in the 3 clusters, and they have they're overrepresented for the corresponding gene, for the corresponding biological association. We also checked whether the genes in the clusters are overrepresented for neurospecific or microglia enriched genes, and we saw that the genes in the cluster which corresponds to the nervous system terms is overrepresented for microglia, and has microglia and neuron enriched genes, and this is in line with recent discoveries about the nervous system. So that was another validation, pretty good validation for us. And the main discussion is that essentially we used 3 different feature vectors to obtain this sort of clustering. Results were quite similar. So the main 3-4 clusters, they respected the topological similarity, and they were with the same biological meaning. So it showed that this procedure provides us with a quite stable way of assigning biological similarity to the different clusters, and a new way to finding gene modules. We tested the popular WGSNA method on over the 105 brain regions and their gene expressions. Of course, this method is known to produce many small clusters with quite high instability, but what was interesting that the clusters, the genes from the second cluster, which is the immunity cluster, which is this one, were in fact not considered, and we know that their expressions are very sparse, or they're expressed in very few regions, and this shows that, well, the WGSNA misses on capturing genes which are sparsely expressed in very few brain regions. So it showed that our method in using networks rather than just plain vectorial representations of genes can bring something new for the discovery process. Future steps, of course, will include discovering more clusters. Clearly, using 3 clusters is just, understatement, but we just wanted to broadly test our hypothesis whether using gene networks would actually make sense, because no one in the literature has so far used to represent genes as separate networks. So it was very important for us just to test would it actually make sense at the broader level. And we saw that it makes sense, so now we will continue with exploring more and smaller clusters so that we can find groups of genes with high topological similarity and much specialized biological function. We'll probably also focus only on neocortex regions, due to well-known facts in the imaging literature. So thank you for listening. I hope that this explains some of the questions that you might actually have had. If not, you can always email me at z.kuncheva@gmail.com, or just find my email at the top of the poster. Thank you for listening. Bye!